Okay, hello everyone. Welcome to the second lecture of the special lecture series. So my name is Min Zhang. I'm professor of statistics and the associate director of data science at Purdue University Center for Cancer Research. So this seminar series is part of our NCI-funded R25 summer workshop on big data training for cancer research. Today we have three panelists, John Fry, Nadia Lama, and myself. So John has helped with setting up the Zoom meeting and handling all the registrations. And he will also record the lecture and post the recording at our workshop website later. And now I would like to introduce our webinar moderator, Dr. Nadia Lama. Dr. Lama is a research assistant professor in the Department of Comparative Pathology at Purdue University. She has been the manager of the Purdue side of the Collaborative Core for Cancer Bioinformatics since 2015. She has a background in biochemistry, molecular biology, and computational biology, and got her PhD in 2015 from Purdue. Dr. Lama's research interests include single-cell data analysis and data integration, with a focus on immunological diseases of the prostate. So with that, I will hand it over to Nadia.
Hello, everyone. Thanks for joining us and welcome. So again, just a couple of housekeeping notes. We will keep you muted, your mics off for the duration of this presentation. Feel free to use the Q&A box at the bottom of your Zoom screen to ask any questions. And if you see questions that you would also like answered, go ahead and upvote them, and those questions will sort of bubble to the top and have a greater likelihood of being answered. And so now I'll hand it back to Min.
Thanks, Nadia. So it's my great pleasure to introduce our speaker today. So Dr. John Quackenbush is a professor of computational biology and bioinformatics, and also the chair of the Department of Biostatistics at the Harvard T.H. Chan School of Public Health, professor in the Channing Division of Network Medicine, and professor in the Department of Data Science at the Dana-Farber Cancer Institute. So John's PhD was actually in theoretical physics, but in 1992, he received a fellowship to work on the Human Genome Project, and this led him through the Salk Institute, Stanford, the Institute for Genome Research, and to Harvard in 2005. So John uses massive data to probe how many small effects combine to influence human health and diseases. So he has more than 300 scientific papers and over 81,000 citations. Among all the honors, like the recognition in 2013, is a White House Open Science Champion of Change. So today, Dr. Quackenbush will speak on why network matters. So please join me in welcoming Dr. Quackenbush.
Thank you very much, Min, and thank you, Nadia and John. As panelists, do you want to have a discussion at the end? Because I just want to see how much time we have. I mean, I'm not going to talk for the whole 90 minutes, but are we going to have a panel discussion at the end?
Not really a formal panel discussion, but more Q&A.
All right, good. I'm all set then. So I'm going to start sharing my screen and hopefully get the right screen this time, which I think I have. All right. So I always hate introductions because it always makes me think about the things I do or don't do. I've been really struggling with large-scale genomic data since I started working in genomics. You heard my background was in theoretical physics. So I started working in genomics in 1992. And a big part of the question we've been asking ourselves is how do we take the diverse data that's coming from projects like the Genome Project and all the extensions that have developed since the Genome Project started, and how do we really understand what drives human health and disease? And over the years, I keep coming back to this idea that the way we have to represent data if we want to make sense of it is to use networks. So I understand the audience here is pretty diverse. And so what I'm going to do is I'm going to take you through at least a little description of how the field has evolved and changed over time and how our thinking really has come around to this idea that networks are going to be a fundamentally important way of representing data. I'll talk a little bit about what we mean by networks, and I'll define precisely what I mean when I talk about a particular type of network, a gene regulatory network. So the starting point for me for a lot of this is the sequencing of the human genome. And as many of you may recall, this project was completed 20 years ago, or at least it was announced complete, with the near simultaneous publication of the public and the private human genomes in issues of Nature and Science. And although it took another few years to really complete and finish the genome, what these two simultaneous papers did was provide us with a reference genome sequence and a reference collection of human genes. And I think many of us were surprised by the fact that there were such a small number of distinct genes found. There were many transcript variants, but we had a collection of about 25,000 genes that we could think of as a reference collection for a human. And if you read a lot of the press releases, everyone talks about having a blueprint for a human being. And that was going to lead us to an understanding of what drives human health and human disease. But if you think about this more deeply, even as soon as I say this, you start to recognize that the problems and limitations with saying we have a blueprint for making a human, let alone a blueprint for making a human cell. And that comes down to really recognizing that there are over 250 different cell types in the human body. Each one of those cell types in your body should have essentially the same genome, yet each one of those cell types carries out a very different biological function. So even though we have the same genome, the parts that are being put together in different cell types and in different ways manifest themselves in distinct functional roles. In the same way, if I look at my liver cells and your liver cells, they're going to be carrying out slightly different functions. They're going to be behaving differently. And so I could argue that the genetics and how genes are expressed and how our traits manifest themselves are going to differ between you and me. And in the same way, if I look at my liver when it's healthy and my liver if I had liver disease or if I had liver cancer, the healthy and diseased cells are going to behave differently. And so we start to recognize that just having the genome by itself doesn't give us the entire picture. We have to think about what genes are expressed, when they're expressed, why they're expressed, and then how things like mutations affect the function of different cells and different cell types. So we've been really fortunate working in this field that the cost of sequencing genomes has just plummeted dramatically. I showed you the first genome sequence in 2001, the publication of these first two genomes. And the estimate was in 2001 that the cost of doing a third genome was something on the order of $100 million. The cost was dropping pretty quickly, and people often compare this initial decline to the decline in computing costs or the increase in computing power, this defined by Moore's law, that essentially every 18 to 24 months, the cost of computing drops by a factor of 2. And that's more or less what we saw up until about 2007, when a new generation of sequencing technologies were introduced. And people use the term next generation sequencing to talk about these, meaning whatever was beyond Sanger sequencing. But what's important is that the cost of sequencing plummeted, and it was dropping by about 33% per quarter. So every 3 months, the cost would drop by a factor of a third. And what that meant was we started to see a real explosion in the amount of data that we were able to generate. We also saw the times for generating data falling dramatically. And so, when I look at 2001, not many of us could afford to have a genome sequence if it was $100 million. By 2009, I used to give talks about the Genome Project, and I would say, look, if my wife or son had a rare tumor, I'd mortgage our house and sequence their genome, because even then we knew enough about genetic variants and their association with disease that we could start to think about using that in a clinical setting to make decisions. Today, the cost of sequencing a genome is down around $1,000. And what that actually means is sort of vague. But the bottom line is we went from unaffordable to almost anyone to I'll mortgage my house to sequence my genome to I'll pay for it on a credit card. And that simple statement really underscores why we've seen such an explosion in the amount of genome sequence data we have, and why we can start to think about taking that data and putting it into context. So I tried to find an estimate of how many genomes had been sequenced, and unfortunately, it's really hard to find. But this is from 2 years ago in April. This was actually the anniversary of the publication of the Watson and Crick paper. And in 2018, the Broad Institute itself had claimed to have sequenced 100,000 genomes. And what that means, I can't really tell you. All I can tell you is that today we have hundreds of thousands, if not a few million genomes that have been sequenced. Some are linked to clinical data and other data. And that genomic data itself provides us with the opportunity to start to think about what the genes are and what they do and what their association is with disease. And that's an important point, that sequencing a genome in and of itself doesn't do anything unless it's linked to other sources of data and information. And this is a place where we've been really fortunate in that, when I started working on the Genome Project in 1992, we were thinking about generating genomic data. As we generated data from more and more individuals, we started to think about the phenotype and the type of data that's included or captured in electronic medical records or other medical records. But today we have a much wider range of data that we can start to draw upon. And that includes data from things like drug research, data from other clinical tests. We have health data that comes from insurance companies and Medicare and Medicaid here in the US that are in some ways distinct from, but complementary to the data in electronic healthcare records. We have the opportunity to get home health data in some studies. My colleague JP Onella has built a whole toolkit for using mobile phones to collect data. And we can even look at interactions between individuals and their self-reported health through social media and through other venues to try to get a sense of what drives human health and what drives human disease. So there's still a lot of unanswered questions, but what it's allowed us to begin to really think about doing in a very concrete way is to look at our genetic background and to begin to do things like to estimate our own individual genetic risk. And we all carry a genetic risk of something, but we'd like to do a better job of early detection. Once we detect disease, we'd like to be able to group patients or group individuals into subgroups based on their own unique manifestation of that disease and how far it's progressed, really with the goal of understanding how these factors, together with things like lifestyle and environment, which influence risk and other things, also influence what the optimal treatment therapy would be, with the goal of improving survival and quality of life for these individuals. Okay, so this is sort of the broad goal in precision medicine, making sense of all of these data. And honestly, we've made a good deal of progress just looking at the individual mutations or changes in how cells express individual genes. And so we have good examples of targeted therapies that target either gene amplifications like HER2 amplification in breast cancer with drugs like Herceptin, but then also in cancers, a number of different single gene mutations that are targeted by individual therapies that are directed toward the proteins encoded by those mutations. And we're not just limited to cancer, although cancer is a place where we have far more data and it's far easier to develop therapies, and we can talk about why later. But there are also drugs, for example, in cystic fibrosis that target specific mutations. And a big part of our challenge over the years has really been finding which of these mutations is linked to which therapy or which combination of therapies. And so in developing therapies, we've also been thinking about developing ways of grouping patients into subgroups and ways of developing diagnostic biomarkers which are a combination of measurements and an algorithm that allow us to assign patients to groups. Now, this presents a very rosy picture, but the truth is that the data are much more complex, much more messy, and that it's actually far harder, even when we have a known mutation that has a drug that should specifically target it, to select a therapy which we know is going to be effective. So this is just showing some data from the CDC. It's a few years old, but it's still representative of what we see, where we can look at genomic alterations and we can look at particular drugs in oncology. And for some of them, the black are the responders and the red are the non-responders. For some of these, we see that almost everyone who tests positive for the genetic biomarker with a simple yes, no as a biomarker, do they have the mutation or not, responds to therapy. But then we have many other instances where the situation is almost flipped, where the number of responders is dwarfed by the number of non-responders. So the question is why? Why, if we have a mutation and a drug that targets that mutation, is our ability to predict who's going to respond so ineffectual? And part of the answer is that biology just is not easy. And there are lots of examples of this. A lot of people have argued that we can just take things like machine learning and feed in all the data we have and arrive at therapies. So this is a story that appeared in the online journal Stat News now almost 4 years ago. But it was probably 10 years ago that IBM, after having its Watson computer system beat Ken Jennings and win on Jeopardy, after Watson won on Jeopardy, IBM sold Watson as the cure for cancer, that they were going to feed in all of the literature on cancer, and they were going to come up with recommendations for treating each individual patient. And when they announced this, it was pretty obvious, I think most of us who looked at this, that this was just not going to be possible. Part of the reason is that what's presented in the literature is often contradictory, it's often incomplete, but more importantly, for what we read in the literature, we don't often have a lot of background about the individuals themselves. And so matching an individual patient where we have very complex data that may distinguish them from other patients with something reported in the literature just isn't possible. So it's a very interesting article if you want to take a look at it. They actually compare IBM Watson to the Mechanical Turk, this chess-playing robot that was built, I think, in the 1700s in Austria, that really wasn't a chess-playing robot. It was a chess grandmaster moving the pieces from under a stage. And in the same way, Watson wasn't able to make sense of this complex data. Part of the problem was, or part of the solution that IBM came up with, was to actually have humans reading the literature and trying to interpret it. But even there, it was difficult. And I would argue that a big part of the reason why Watson failed was that the problem was underspecified, but also that we lacked an underlying model that would allow us to interpret the data in a meaningful way. And to me, this is really interesting because this is not a new problem. So this is a paper from 1997 by two computer scientists who at the time worked at Microsoft on the problem of ensemble classifiers, David Wolpert and William McCready. And they wrote a paper on what they called no free lunch theorems for optimization. And I love this paper because if you read it, and I'm just going to excerpt the introduction down here in the lower left, if you read it, it sounds like an indictment of almost every machine learning paper you've probably read in your life. The past few decades have seen an increased interest in general purpose black box optimization algorithms that exploit limited knowledge concerning the optimization problem on which they're run, right? Wolpert and McCready were looking for a universal weighting function for ensemble classifiers and couldn't find one. So they wrote this paper, and in this paper, what they prove are a number of what they call no free lunch theorems that demonstrate the danger of comparing algorithms by the performance on a small sample of problems. right? Something not only people doing machine learning are guilty of, but people doing statistics are guilty of as well, right? You have a small problem, you prove, or you have a simulated data set and you prove something works. And when it meets real data, it falls apart. But the other thing that really struck me, and I read this paper first in 2002, and this has been a part of the way I've approached looking at biological systems, is the second sentence, which is highlighted here. The same results also indicate the importance of incorporating problem-specific knowledge into the behavior of the algorithm. And so for me, this was really the fundamental revelation that led me to think differently about how we approach problems. And one of the things that I'll argue is that networks represent a biological constraint that we can impose on some of the algorithms that we use to try to model biological systems and that we use to try to make predictions and interpretations. So this comes back to this problem I posed earlier. What makes cells different, right? If we have cells in our body, they all have the same DNA. The DNA is transcribed to RNA and then translated to make proteins. That's the central dogma of molecular biology. And I would argue that a big part of what drives this is the process of turning DNA into RNA. And I would argue that this process is actually driven by and constrained by gene regulatory networks. And that part of what makes one cell type different from another cell type is that each cell type has its own unique regulatory network. All right. So we just had a grant review come back from NHGRI where we proposed network-based methods. And one of the referees, two of the referees loved it, one of them hated it. And the referee who hated it said, well, this is obviously wrong because there's only one network. And what we're proposing to do is to identify disease-specific networks. I think a lot of this is semantic, but I think you can make a very persuasive argument that networks are a fundamental component of biological systems, and that these networks really do differ between biological states. So we have a long history of representing complex data in biological systems using networks in a network formulation. When I say network, what I mean is actually something very simple. A network is a way of representing complex association patterns between various entities. And in that sense, you can represent a network in a lot of different ways. And one of the things that makes a network really easy to compute on is the fact that we can represent a network by what we call an adjacency matrix. So I've got a little toy network here on the left. There's 6, call them genes, represented as nodes, and some kind of connection between them, which I will call an edge. So we have nodes and edges, and the edges represent some kind of an association. So this could be correlation, this could be regulation, it could be almost anything. But if I just look at this diagram, 1 is connected to itself, it's connected to 2, and it's connected to 5. And if I look at the first row of this matrix, I have a 1 in position 1, a 1 in position 2, and a 1 in position 5 to represent this kind of connection. And you'll notice that this is symmetric around the diagonal, so I have the same thing for the columns. And then if you pick any other gene, let's say row 3, it's connected to 2 and 4. So gene 3 is going to be connected to number 2 and number 4. So I've got a nice way of representing a network as a matrix called an adjacency matrix. And how I populate this, this matrix, or how I represent this network, is going to depend on what kinds of interactions I want to try to model. So they can be abstract interactions, right? Like correlation and gene expression profiles. They can be physical interactions like 2 proteins measured to bind together, or a transcription factor measured to bind to a cis-regulatory region of a gene, or they can be almost anything else that you can imagine, right? A network is simply a way of representing associations. So what we try to do is we try to define our networks in a very precise way when we talk about them. And the way we think about networks in the kinds of instances I'm going to talk about today are in comparing patterns of gene expression between different physical conditions. So imagine I have gene expression data where I have matrices that contain genes for individuals or individuals representing different conditions. And I'm going to separate these into two phenotypes. And if I have my two phenotypes, one of the things we've been doing for years is looking for differentially expressed genes by using some kind of variation on a t-test or a linear model to identify those genes that are different. But what I would argue that we also want to do is we want to try to infer the regulatory network or networks that are represented in these data. And again, the semantic difference between one network or many networks is kind of small. I'm going to refer to networks for phenotype number 1 and a network for phenotype number 2 as being distinct. But what we can do is we can imagine looking at the topology and structure of those networks. So there are patterns of connectivity in the network which are distinct. There are also community structures in those networks. And I'll touch on that briefly toward the end of my presentation. But essentially, when we look at community structures, what we recognize is that there's subgroups and networks that are connected together, right? We all have mobile phones. We can call anybody in the world, but we don't. We call our family, right? So I'll call my wife will call my son will call his grandmother will call me, right? And so on any given day, any two of us are more likely to call each other than we are to call some random person in the outside world. And while I've enjoyed talking to Min about presenting here, on most days, I'm not going to call Min, while almost every day I call my wife. So what we can see is that we've got patterns of association that represent my family community compared to the outside world. And what we find is these communities change over time, right? We have a work community, we have a social community, we have a family community. And if I look at your patterns of calls, or text messages, I can probably predict whether it's a weekday when you're working or a holiday when you're going to hang out with your friends, all right? So the structure and topology of the networks matter, and differences in those structures and topologies matter. And if we take gene expression in these topological differences, we can often see gain insight into changes that actually tell us a lot about the driving features of the biology. And what we've increasingly tried to do is to represent these networks as individual specific networks, where we can think about the networks themselves being biomarkers for particular traits or features of the networks being biomarkers. So you heard earlier that I was recognized as an open science champion of change in 2013. I got invited to the White House and we thought we were going to meet Obama, and he got called away, so we never met him that day, but still had a very nice day. But I'm really committed to open science. So this is one of the things we've tried to do. We've collected all of our different methods. And if you look here on the right-hand panel and read the names, you'll start to notice something. All of our methods, Monster maybe is a little side trip into cryptozoology, but every one of the methods that we've developed is named after an animal. And so we call our collected package of network methods the network zoo. I can talk about individual ones. Today, I'm going to be talking largely about panda and lioness, but they all have implementations in R, Python, MATLAB, and C. And Marwan Ben-Gabila has really done a good job of collecting them together, developing a whole set of tutorials and Jupyter Notebook tutorials that we call netbooks that provide access to the underlying methods. So if you're interested, these methods are available. And what I'm going to do is I'm going to talk about how we use these methods to address very specific problems. So I'm going to frame this in the form, I guess, like Jeopardy in honor of Watson questions. Question number 1, can we model the gene regulatory process? And a lot of this goes back to what I talked about earlier with us trying to impose biological constraints on our modeling, right? Use what you know to constrain these large, complex, hard to fit models. So I'll start with what we understand about gene expression and gene regulation. So I'm going to imagine that if I have a gene, the output of the regulatory process is an RNA transcript that I can measure. When we started this years ago with microarrays, increasingly that we can measure using techniques like RNA sequencing, but I can measure how many RNA transcripts I get from a particular gene. How do I make those transcripts? Well, biologically, we know that that's because an RNA polymerase binds and copies that RNA. But we also know that that process is facilitated by the binding of transcription factors that can either open the chromatin, help assemble the RNA polymerase II complex and allow it to bind. So they can facilitate transcription or they can inhibit transcription. And so that's going to be our model. And when we started this whole process of trying to build these models, the very first method that we developed as part of our zoo was a method that we call PANDA. And this was largely work that Kimberly Glass did as a postdoc working with me. I forget when we published this, it's about 8 or 9 years ago. Kimby's now an assistant professor at Harvard Medical School and Brigham and Women's Hospital. But PANDA is really one of the core methods that we've developed and used over the years. And PANDA is based on kind of a simple approach. It's something derived from communications theory, which I'll show you in a little bit, called message passing. And the basis for PANDA really comes down to our understanding of what the regulatory process entails. That we have transcription factors, which I'm going to represent by these blue boxes or these different color boxes. And these transcription factors are going to bind to the region of genes and regulate their expression. So the boxes are the transcription factors and the circles are the genes. And for example, this blue transcription factor, I would say, has some evidence that it regulates gene number 2 and number 3. The yellow transcription factor only seems to regulate number 2, and the green transcription factor regulates 1 and 3. All right, so that's going to be our basic model. And we call this a bipartite graph because there are 2 components of that graph, the circles and the squares, or the genes and the transcription factors. So this is our fundamental regulatory network. And what we do, in addition to guessing that this is the network, is we also recognize that the network itself tells us something. That if I look at the proteins, the transcription factors, one of the things I know is some of these proteins interact with each other and bind together to form transcription factor complexes. So I can look at protein-protein interaction data to see likely complexes. But the other thing, which I would assume, is that if 2 genes are regulated by the same transcription factors, they're likely, these 2 are likely regulated by the blue, they're likely to be correlated in their patterns of expression. So we can start to look for correlation patterns of expression as evidence of regulation. In the same way that we can look at protein complexes as evidence of regulation. So what we do is we try to look at these diverse data sets and we try to seek agreement between them using this idea that was first developed in communications theory called message passing or affinity propagation. So this is based on the simple idea that in any communications process, there are always 2 participants. There's a transmitter and a receiver. So what I'm going to imagine is that I am going to talk about this method today. And at the end of my presentation, I'm going to start to pick on John, but I'm going to give John a quiz. And we're going to see how well John does in that quiz, right? And if John listened intently to my lecture, and if I did a good job explaining this method, then John is going to take the quiz and he's going to get 100%, okay? However, if John fails the quiz and you didn't attend my lecture, all you knew was that he failed, you would have no way of knowing whether it was because I did a horrible job of explaining it or he did a bad job of listening, right? So how do we resolve that? Well, one of the things we do is we recognize that both these parties have to be engaged in the process. I'm responsible for communicating. The transcription factor is responsible for sending a message to its target, but the target has to be available to receive that communication or to respond. Now we can define mathematical functions and try to fit these, but the truth is that if we don't see anything but the output of the interaction, we can't estimate these functions. However, if I were to give everybody listening to this lecture the same quiz, if everybody failed, I did a bad job. So we know I'm the offending party. If everybody passes, everybody did a good job. If everybody passes but John, then I did a good job and most of you did a good job. And we're just all very disappointed in John for not paying attention. Okay, so we can start to see where communications might break down if we have a big enough ensemble of data that we can use to estimate these functions. Okay, so how do we really do this in the context of the no free lunch theorem? Well, we want to start with a guess. So we're going to look at transcription factor motifs and map them back to the genome. There are lots of subtleties involved with that. We're constantly trying to do it better, but we start with an initial guess as to what the network looks like. Then what we can do is look at those other sources of evidence for regulation, protein-protein interactions between transcription factors, largely from the string database, and then co-regulation, which really comes from looking at correlations in gene expression that are deduced from each one of our experimental or clinical models. So what we can do is we can take that network, and for each edge, we can calculate a responsibility and availability. If one of these goes to 0, we can gets near 0 below some threshold, we can update our model. And then we just repeat this process over and over and over again until the network models converge. So we've applied this basic paradigm in lots of different situations. The original Panda paper has some examples. One of my favorite examples is an analysis we did of 2 subtypes that we'd identified in ovarian cancer. And essentially what we did was we looked at these subtypes and we built 2 sets of network models. And we asked, and this was one of the very first large-scale comparative network analyses we did, we simply asked, what are the differences between these individual networks? And I like to talk about this example because for me, it was really transformative. That when I started thinking about the networks themselves, the genes were always the same. We always input the same set of genes. We always get the same set of genes out. The reason I claim that the networks are different is that the atom of the network in the analysis we did are the edges. And it's the edges that differ between one network and another network. And it's the pattern of connections between the nodes, these edges, that really tell us a lot about what's driving the biology. So without boring you with all the details, we compared 2 subtypes of ovarian cancer that differ by their potential to generate blood vessels. So we have an angiogenic and a non-angiogenic subtype. We built networks and we looked at the 10 transcription factors that had the biggest differences in their connectivity. So they're all represented here with, I believe, the red being angiogenic and the blue being the non-angiogenic. And I apologize if people can't see the colors easily, but here's the angiogenic and here's the non-angiogenic. And if I flip back and forth between them, you can start to see differences. But the bottom line is that we see differences between them. And then when we started to look at where those regulatory differences were, what we saw was that it was combinations of transcription factors that were regulating things that changed between one state and the next. And so we went back and we looked at the literature and we asked, all right, what do we know about transcription factors? And one of the things we know is they form specific complexes that we saw being overrepresented in changing their targets in the data set were complexes that we had evidence for and support for in the literature. And that these complexes, in fact, were known to affect the process of angiogenesis. And what was even more compelling is that drugs that inhibited the formation of these complexes or compounds that inhibited complex formation were actually found to inhibit the angiogenic process. So this was a really exciting discovery for us because what it pointed us to was this idea that the networks themselves were telling us something about the potential drivers of these different phenotypes and how you might convert one to another or how you might interfere with the process. Because one of the things we know is that if tumors are creating their own blood supply through the process of angiogenesis, that if they're creating this blood supply, the outcome for those tumors is actually much worse. So if we can interfere with that process, we can actually potentially improve outcome for patients with disease. So we've actually published a number of papers now that capture the kinds of findings we saw. This is the angiogenic, non-angiogenic ovarian cancer comparison I just talked about. This is published now 6 years ago. But we've looked at other things like sex differences in chronic obstructive pulmonary disease. We had a project that we did in collaboration with someone who was a postdoctoral fellow at the time at the National Institutes of Health, but doing a master's degree at the Harvard School of Public Health. And we have a whole bunch of other papers that we've published using this kind of approach. But one of the things that I started with was this idea that we all have our own individual networks. And the papers I just talked about were some of our early attempts to build a network for one phenotype and a network for another phenotype, and to ask, how do those networks differ from each other? But as we started to look at this problem, we started to really recognize that each individual within a population potentially had their own networks. So how do we move from this idea that we can take multiple samples in a population and build one network for that population, and maybe for a different population build a different network that they can compare? But how do we recognize that each person, each sample in this original data set likely has their own unique network? And so we ended up moving from this idea that we could aggregate networks by looking at a population to thinking about the fact that this aggregate network represents input from all the individual samples, and that what one could do is use the available data to infer individual sample networks that on average gave us this single sample network. And so this is a method that we call Linus. It's a method based on linear interpolation. And it was actually discovered quite by accident by a summer student. We had a postbac student, Michael Tung. And what Michael was doing was trying to estimate confidence intervals on all of our network edges. And to do that, he was doing a resampling analysis. And we had originally told Michael that what he should do is just leave out one sample, leave out 10% of the samples, do the analysis, put those back in, leave out another 10% at random, and repeat this many times to get estimates. What he did was he actually ended up inferring a network for all of the samples and then leaving out one sample. And even in leaving out one sample, it's not a big surprise. But what we recognized was the networks were subtly different. And so if I look at these networks and I compare them, the network with a sample and the network without a sample, what we discover is there are differences, right? So if you can imagine that in doing this analysis, we left me out. If I had one edge that was much weaker than everybody else's, leaving me out would decrease the estimate of that edge. And so if I look at the difference between the network with me and the network without, if I have one edge that's much stronger than everyone else's, leaving me out is going to decrease the estimate of that edge. And so I look at the network with me and the network without me, what I'm going to see is evidence of the place where my network is stronger than everyone else's. And so it's pretty easy to imagine that if I take this difference and scale it by the number of samples and add it back to the network without me, I'm going to get an estimate of my own individual network. And so you can imagine doing that for me and then putting me back in and then taking you out and then putting you back in and taking the next person out and on and on and on. And if I repeat this process over the entire population, I'm making an estimate that to first order, these network edges are linear sums or linear functions of all the different connections that we see. But it's very easy to write out a simple mathematical function for calculating these network edges and then running the process iteratively across a large collection of data. And so that's what we did. When we tried to publish this paper, the paper was actually in review for more than 2 years before it got rejected by the first journal we sent it to. And the referees kept on asking us for more and more evidence. We actually went and analytically proved that you could do this for Pearson correlation networks, that you could make this estimate for mutual information networks. We showed by simulation that you could do it for other types of networks where there wasn't an empirical solution. And we had pages and pages of proofs. And one of the referees said, well, I understand the math, but I just don't feel that this is right. And so the paper got rejected. Poor Kimby and Marika Kudra, who worked in this, they were beside themselves because, and Kimby put it really well, that mathematics is not about feelings, it's about proofs. And we had proved that this works. So we eventually got the method published. And one of the things we did with this method was we looked at a very simple example. And this was just looking at data from the yeast cell cycle. It's a canonical data set in which yeast was taken through 2 cell cycles. It was done in duplicates. And one of the things we know is if we look at the gene expression data, it's actually correlated. And here it's shown as a correlation or anti-correlation because the experiment used 2 different fluorescent dyes. So correlations look like anti-correlations. But leaving aside the dye, one of the things we did was we took each one of these samples, and the first data set, we calculated individual sample networks. We took each one of the samples in the second data set and calculated individual sample networks. They were all taken at the same time, so we had pairwise sets. And we looked at the correlation in the networks. And what we saw was the networks themselves were highly correlated. So that was really interesting. If you look at the most variable edges, and now we're showing the paired time points side by side, what you can see is that high level of correlation, but you can also see an oscillatory pattern. And given the fact this is a cell cycle experiment, it's not surprising. When we looked at the data, what we saw was that the edge weights shown in black here were oscillating in our models. And the most variable edges were associated with 4 transcription factors that were known to regulate the cell cycle. But the other thing that we saw was that their target gene expression levels shown in red were also oscillating. And we first saw this, we got very excited. Then we got very disappointed because the frequency of the oscillation of the edge weights was about twice the frequency of the oscillation of the target genes. And then we sat and thought about it for a day and came back very excited because we recognized what we were seeing was message passing working fully. So a signal to turn on, turn off, turn on, turn off, turn on. Exactly what we should have expected from this method. And in fact, there was then chip data on these cell lines or on similar yeast in synchronization. And we saw binding of the transcription factors that was very well predicted by using Panda and Linus. So we've looked at other situations. And I'm actually going to skip this slide because we're going to come back to glioblastoma later. I'm sorry I didn't take it out. So I'm going to put this aside for a minute, but I'm just going to say that we've applied this method extensively now. So we published a paper looking at GTEx release 6, in which we compared cell lines to their tissue of origins and found differences when you make cell lines. We looked at tissue-specific regulation in 38 different tissues and saw distinct changes in regulatory patterns that activate communities of genes associated with particular cell functions, as you might expect, given the fact that 38 different tissue types have different cells that carry out different functions. One of the things we're really proud of is looking at colon cancer and looking at the differences between males and females and starting to understand, this is sexual differences between males and females in 29 different tissues. But then also looking at sex differences in colon cancer, where we can explain clinical features like the fact that males and females respond differently to chemotherapy, something that we have no evidence of the mechanism for based on gene expression, but which we can find distinct differences in the regulation of drug metabolism pathways in surgical samples before chemotherapy that tell us that males and females are going to be likely to respond differently to chemotherapy. And in fact, that tell us that even among the females, those who are more male-like than the others in the regulatory processes behave more like the males than the rest of the females. So there's a lot that we can see in looking at gene regulatory networks, which really leads me to the question that was the title of my talk, right? Or the basis for the title, why do networks matter? And I will tell you that we debated how to really present this for a long period of time. Debbie Weghill is a postdoc who works with me. Debbie actually had an opportunity to move to University of North Carolina, where she's applying for a K99R00. She's working with Jen Ye there, who studies pancreatic cancer. And what we did was we took a look at gene regulatory networks in health and disease, using pancreatic cancer as a model, to try to understand what additional information the networks were giving us in a really clean sample. And Debbie started with a little toy model. So this paper was just published 2 months ago. Debbie started with a really nice little example. It's a toy model. And part of what we were arguing was that it isn't just gene expression by itself, but it's other things like correlations in expression that give us insight into disease. And so she had this little toy model in which we had 2 sets of individuals, so a group of 9 healthy individuals and a group of 9 individuals with disease. We had only 4 genes, but the genes differed in their patterns of expression. So we can find differences in expression that distinguish we have patterns of expression that don't exhibit any differential expression between the healthy group and the disease group. So here's gene 1 and the healthy and disease group, no real difference. Here's gene 3 and the healthy and disease group, no real difference. But if we look at co-expression, if we look at correlations in the pattern of expression, what we see is there are distinct differences in the pattern of co-expression that distinguish the healthy individuals from the diseased individuals. And that's something we've always known, that correlations in expression can change between states. But we tend to shy away from looking at just correlations. And there are well-established methods like WGCNA that look at correlations. We don't consider that a regulatory network. We would call that a correlation-based network. And there are clearly things you can see from looking at correlation. But we ask, even if we find something in correlation, can we find something that comes back to our understanding of this regulatory process, right? And I talked about this earlier, and this is one of the figures from Debbie's paper now, that if we look at transcription factors regulating genes, that co-expression, which we know can differ between states, is some evidence for regulation, but it's not conclusive. We can also look for protein-protein interactions, as I mentioned earlier, which is some evidence for regulation of particular genes by sets of transcription factors. And so we can get additional insight into the regulatory process. So what we did in this Frontiers in Genetics paper is we took pancreatic cancer data from the TCGA. There was RNA-seq data available. We used Linus and Panda, the methods I talked about in the context of yeast, to infer individual sample networks. And then we just did a basic comparison between 2 subtypes, the basal subtype and the classical subtype. And in the same way that we would do differential expression analysis on the gene expression levels, we looked at the edge weights and we did differential targeting analysis using the same tool we used, Limma. So we looked at our differential regulation. We also looked at differential expression and co-expression, and we did a gene set enrichment analysis. And so here's the result. This is our regulatory targeting based on Panda and Linus. This is the data from co-expression, and this is the data from expression by itself. And it's really interesting. You see some overlap between all 3, but a lot of overlap between correlation and our differential targeting, our differential regulation analysis. And this really isn't surprising because one of our assumptions is that correlation is actually evidence of regulation. we do see things, some genes that are differentially expressed that we don't have good evidence of differential targeting for. But these are largely things which aren't very interesting in the context of understanding pancreatic cancer and these two distinct subtypes of pancreatic cancer. But what's really interesting is when we look at what the differential targeting, the differential regulation tells us that nothing else does. What we see is there are a whole host of immune-related processes, epigenetic processes, and cell cycle processes that were completely missed using these other approaches. And all of these, if we start to dig into the genes that are involved and look at the individual processes themselves, really point to clinically observable differences between these two subtypes. Now, all the subtypes in pancreatic cancer are bad, all the outcomes are bad, but this actually provides some evidence as to why some individuals respond better, for example, to immunotherapies in pancreatic cancer than those who don't. So the regulatory processes are actually giving us insight by building these regulatory networks that we couldn't have found looking any other way. The other thing I just wanted to mention is this glioblastoma example I had mistakenly left in earlier. This is work that was done by Marieke Kujer, and this is a paper that was just accepted recently as well, in which what we did was we took individual data from TCGA on glioblastomas, we inferred gene regulatory networks, and we divided the patients into two groups depending upon survival. And we compared the networks for the long-term and short-term survival patients. So long-term and short-term in glioblastoma is kind of relative since survival tends to be very bad. But we looked at these different groups, and what we did was we asked, what are the regulatory processes that distinguish the better outcome from the worse outcome groups? And what we found, again, was that we have differences in the regulatory processes that are reflected both in genes that are differentially regulated, but also in communities, in two different TCGA data, or two different data sets, in communities that are involved in things like immune response. And so what we're starting to see is evidence for the regulatory processes in hard to treat diseases like glioblastoma in the brain, where the blood-brain barrier would suggest that immunity doesn't play a huge role. We're seeing differences in these immune processes, which actually distinguish patients who do well from those patients who do poorly. What's even more interesting is that when we start to look at the community structure, what we see is that there are genes which are at the cores of their community. So when we talk about networks, we often talk about hubs. One of the things we find is the hubs, the most highly connected things in the networks, often have very little explanatory power for telling us anything about the nature of disease. And part of the reason we think that's true is that if something is too highly connected, you can't perturb it much without having catastrophic consequences for the cell. But if we look at a functional community and we look at the core of that community, or the modularity that's carried by a gene at the core of its community, we often call that the core score, we can identify what are local hubs. And those local hubs we find are actually genes which are known to be heavily involved in the development and progression of diseases like, in this case, glioblastoma, that the real drivers of disease are driving disease not just by nature of their own mutations and expression, but actually their regulatory effects on other genes which shift processes to alter the phenotypic state of an individual cell or individual tissue. When we look at the pathways that are targeting, again, what we see is a great deal of difference in processes that are associated with immunity and immune response. such that immune response appears to play a substantial role in predicting the prognosis of patients in glioblastoma, and that repressing PD-1 signaling improves overall survival. So if you're interested, this paper is up on bioRxiv. I forget, it was just accepted a short time ago, so I don't think it's appeared in print yet, and I actually forget where it was accepted. But if you're interested, you can find this paper on the bioRxiv. And this is just part of a bigger family of papers that we've published, and I apologize for not taking these out. But why bother with networks? Well, in numerous applications, we've found that networks provide insight into disease that's not found using gene expression or co-expression by themselves. Differential targeting captures not only a phenotype's current expression, but also its potential to respond. What's interesting in a lot of the different examples that we've seen is that if we look at surgical samples that are removed before chemotherapy is used to treat the patients, if we look at those surgical samples, we don't see a lot of difference or variation in gene expression that explains phenotype. But if we look at the regulatory processes, we can actually predict differences in phenotype like response to therapy, because the cells are set up to respond differently in the regulatory networks, whether or not those networks are actually active yet or not. And so I think this is a really important insight, that these networks are telling us something beyond what we can see from just looking at expression alone. We've also published a number of papers using another method called Condor, which looks at the association between genetic variants and gene expression using EQTLs. And we looked at cancer-related genes. There was a paper we published, I think in the British Journal of Cancer, that found that genetic variants that we know increase the risk of cancer are actually regulatory SNPs that regulate oncogenes and tumor suppressor genes, but also a community of other genes associated with cancer-related processes. So these networks and their structure are actually telling us a lot and giving us insight into the development of disease progression and response to therapy. And one of the things we've seen is that changes in network structure can help identify drivers of disease and therapeutic targets. So this last piece I haven't talked about, I'm just going to refer you to something that has now been submitted to Nucleic Acids Research. It's a database that we've assembled of now, I think 120 or 130,000, I'm sorry, almost, I don't know, over 20,000, it's way more than that, gene regulatory networks that we've built. It's a database that we call GRAND, and so the URL is down here at the bottom. What GRAND does is allow you to search for networks that are like your networks if they're derived using Panda. But built into GRAND is actually an idea that came from our analysis and came from the availability of data from the connectivity map and the CLUE project that looks at differential expression of genes in cell lines before and after treatment with therapy, with different compounds. And just like the connectivity map allows you to look at differences in gene expression to predict drug response, what we've found is you can actually do a better job of doing that by looking at differences in network structure. So in this paper describing GRAND, which, and I'm sorry I don't have the reference for it, but I can get it for you, is up on bioRxiv. One of the things that we can see is that these regulatory networks actually allow us to predict potential drugs and drug targets. One of the examples we use in our paper is looking at colon cancer, and we identify a drug that's a candidate therapy for colon cancer that was actually found to inhibit Aurora A kinase, a marker of proliferation in non-small cell lung carcinoma, that we would predict in combination or alone would also be beneficial for treating colon cancer. So the networks are giving us insight. We're trying to build tools and techniques to make this accessible. All of the methods I've talked about today are available as open source tools through the Network Zoo. There are detailed tutorials in a resource we call netbooks, which you can find by going to the zoo. And we've published lots of papers that I think are really starting to establish this approach of modeling networks using prior information about how the regulatory process plays itself out, that doing this kind of informed modeling can really give us deep insight into the drivers of disease. So since this is a statistics based workshop, I'm going to say that there are a whole host of statistical questions that we're still trying to address about networks, how we deal with continuous edges and even defining things like network parameters, betweenness, centrality in the networks, degree in a network. It is something we wrestle with quite a bit. How we find community structure in networks with continuous variables is important. How we estimate network edge strengths and our confidence in those is an ongoing question. There are a whole host of questions around the statistics of networks that we're still working to address. And we're also still working to try to turn these into predictive models in a robust way rather than descriptive models. But I hope, if nothing else, I've convinced you that at some point, looking at networks is going to give you insight that you couldn't get any other way. So I really want to thank Min and everyone else for the opportunity, giving me the opportunity to speak today. I just want to say that this work was really supported by a large group of individuals. This is my current group of postdocs, almost all of whom have gone now to find other jobs. But my research group runs like, now I say it's a neo-socialist collective, because a lot of my former postdocs are still members of our extended group. We meet every Tuesday morning. So Marieke Kujer is at University of Oslo, Mega Patty is at University of Arizona, Kimby Glass and John Plattick are both former postdocs at Brigham and Women's. Abby was one of Kimby's postdocs, who's still part of our extended family. Dawn is a collaborator who's really driven a lot of the work on sex differences with Camila Lopez Ramos. Marwan is still with me. Rebecca's going off to Max Planck in September. Sheila just, I think, took a job at Regeneron. Debbie moved to University of North Carolina. And I've got 4 or 5 new postdocs finally who are starting with me, as well as a couple students. So I really want to thank them for all of their work. And I want to thank you for spending a warm summer afternoon, at least warm here in the Boston area, with me today. So thank you.
Thank you so much. So I have, let's see. I mean, while we wait and see if anyone else has questions, I have a whole list of questions, so I can just, and many of which are just sort of practical. If attendees, for example, want to run, run use some of these network methods, even in the Jupyter notebooks that you provide. So I know you have an AWS, you have this on an AWS bucket, but for students who maybe aren't comfortable with that or just learning, are there any plans to put this on, for example, like Galaxy? Or if not, how much CPU and RAM do you know how much? I mean, could they do something like this, kind of quickly on a, like, I don't know. I'm just trying to see how easy it would be for some of our attendees to run this.
So, you can download this and run it on your laptop, right? It depends on the size of the data set.
Yeah.
So, if you have 10 or 20 or 30 samples, you can run this on your laptop and it's easy. And especially a lot of the early studies we published, they run on laptops. So I talked about doing a couple analyses of GTEx. When we did the GTEx analysis, we had 600 individuals. There were almost 40 different tissues. And if you do the math, you realize that's 24,000 potential sample individual combinations. It turned out there were about 19,500 because not everybody had samples for every tissue. We first ran bulk networks for each tissue, and you could do that, by separating the data out on each sample or each tissue. And it was 600. It was a little slow, but you could, run on your laptop. It wasn't so bad. You could do it in a couple of days. As we went to running all 19,000 networks, it took us, when we first started doing it, it took us 6 months on a big compute cluster. Okay. And it was just incredibly painful. So we submitted that sexual dimorphism paper, and one of the referees complained about how we handled the X and Y chromosomes. So we had to do it again. So the first one took 6 months. We'd optimized the algorithm the second time, so it only took us 6 weeks. And that actually led Marowen to build a GPU implementation, right? So if you have a huge data set, there's actually a GPU implementation. So you can find it and you can run it in near finite time. It really depends on the question you're asking and the size of the samples.
No, that makes perfect sense. And then that also gets to my second question, which is also pretty practical. But so I saw, like, in the example you did on the yeast cell cycle project, you had, I think, 24 samples per replicate or something. So is there like a good rule of thumb for how many replicates and samples you'd feel confident running this on? I mean, I imagine probably not like 3 in each condition. So,
it's hard to come up with a rule of thumb, right? Because in yeast, it was 2 replicates that we were comparing, but it was 24 time points. Okay. And the bottom line is, with those 24 time points, we could infer 24 individual networks, because the network inference problem is sharing information between the different networks, right? So if I pluck out 30 minutes, I'm going to be using information to estimate the network without that 30-minute timepiece in, right? And the difference with and without 30 minutes is going to tell me what things have changed in that 30-minute sample. And it's really based on the idea that each edge weight is kind of an average across all the edge weights. And so if I perturb the system by leaving a sample out, I can interpolate back to try to find the missing edge weight.
Okay, yeah. And well, then how does it handle like unbalanced data between classes, or like if you want to compare networks between like cancer and normal? A lot of times it's hard to convince folks to give normal tissue.
So you can infer those networks. Once you have those networks, then you have the standard class comparison problem that exists in every single cancer study you try to do. And so we haven't solved that, but we've solved some of the other problems. So let's see.
You have a long question. You might want to read it yourself.
All right, I'll try to read it in a minute, but I'm trying to find some. I'm trying to do more sharing. And for some, I think this is, yeah, here we go. Good. So am I showing you the network zoo?
Yes.
Okay, so this is actually our GitHub repository, and there are lots of tools and resources available. One of them is netbooks. And so if you come here, right, there's actually a whole host of little vignettes about using each one of these. So again, you can run these here on the server and it'll take care of it for you. If you go back and want to set up any of these, you can download them and run them locally. If you really want to do them at a larger scale, you can do them using by getting somebody to help you set them up on a local cluster. And there are also GPU versions of all of these. So we try to make it available. All right, so now I'm trying to look at the questions. There are a couple long ones. Oh, I read, somebody who's anonymous said it was a fantastic talk, so we'll stop there. And I don't want to forgive you for having a baseline understanding, because that's the same thing I have. I've just been doing this for a long time. So if you already understood it, you wouldn't be here to hear from me. So that's great. The question says, you talk a little bit about transcription factors being controllers of the regulatory networks, but I've heard other research suggesting that gene regulation is more dependent on open chromatin than the specific transcription factors that bind. Okay. And that's actually a really good question. Should we look at regulatory networks as a product of epigenetic regulation rather than transcription factor function? Absolutely fabulous question, right? You can also talk about other regulators like microRNAs, and we've been trying to capture all of those. So if you look at our collection of zoo animals, one of the animals is puma. And Marieke Kuger developed this. Puma is panda using microRNA associations. And it's panda, but it has an additional regulatory piece layered on top of it. And that's negative regulation by microRNAs. So that is important. But if you look at open chromatin, the open chromatin is a great source of information because you're right, as the chromatin opens up, you have the opportunity for transcription factors to come in and regulate. And people often ask, why did you invest so much time in transcription factor regulation? And the answer is always because that's where the data was, right? We had gene expression data. There wasn't a whole lot of relevant ATAC-seq data. Abhijit Sadawani, who's now an assistant, I think, a lecturer, assistant professor, instructor, assistant professor, I forget which, at Brigham and Women's. He was a postdoc. He and Kimby, along with the rest of our group, developed a method that they call SPIDER. And SPIDER accounts for open chromatin based on ATAC-seq data. So it combines the best of both of these models to try to tease out the appropriate regulatory processes. And we're trying to incorporate that into the models going forward. We don't have a lot of data and experience with it, only because there aren't a lot of data sets we can run it on, right? And not a lot of data sets with clinical information at the end of the day. So, if you want to catch fish, you got to fish where there's water and fish. So, you can't fish in the bucket behind your house. So we try to find good data sets where we can actually link this to clinical outcomes. The other thing I'll toss in here is that John Plattig and Debbie Weghill worked on a method that we call EGRIT. And EGRIT actually takes this whole process in a different direction by incorporating genetic variants in the putative regulatory regions. So you can look at the regulatory control regions where transcription factors bind, and you can actually build individual specific networks based on genotype. And that's another really exciting direction where if we could incorporate open chromatin and other data, it would be fabulous. We just don't have data sets with all the different pieces. So I hope that was a good answer to a far too long an answer to a really fabulous question.
Thank you. And we have another question in the chat. So thank you so much for your presentation. Very informative. Have a question in terms of helping identify what drives disease and subsequent identification of therapeutic targets. What types of changes in network structure, for example, can be implemented?
So again, it's an absolutely fabulous question. And I will tell you, all these questions you're asking, like if you came to our Tuesday morning group meeting, most weeks we'd be arguing about at least one of them, okay? And I can tell you all the things we argue with about PANDA, the associated family of methods, like what is it really telling us about binding and regulation? But if we look at the features that drive the phenotypes, there are three primary features that we focus our analysis on. And I will tell you that the only reason we do that is because there are things that we can easily compute and compare. So one is just the addition or deletion of edges, right? So what edges have been lost. And one of the first examples of looking at sex differences in disease was a study which is still unpublished. And I can tell you why it's unpublished. There's one data set we can't make sense of that disagrees with all the others. So we have a set of consistent data sets and then one oddball. But it was looking at sex differences in Alzheimer's. Okay. And Alzheimer's is a great example of sex differences in disease risk. Females have twice the lifetime risk of developing Alzheimer's, even correcting for the fact that you're likely to outlive me being female. Even if we both carry ApoE mutations, you will have twice the lifetime risk of developing Alzheimer's that I do. Okay, so there's clearly a sex-driven component to this. If we leave out the one data set we can't make sense of and look at the regulatory edges that differ, just taking the edges that appear or disappear, what we find is that there are differences in edges, and that these are genes, these are transcription factors that bind to genes that have estrogen responsive elements. Okay, so it's not that surprising that differences between males and females in disease risk for Alzheimer's might differ in brain-specific genes that are, and it's actually brains and apolipoprotein metabolism that respond to estrogen or testosterone. Okay, they're actually hormone responsive elements. So, you know, those edge differences can be informative. Since we're dealing with a continuum of edge weights, most of what we do is we look at differential targeting. And so we look at differential out degree for the transcription factors and differential in degree for the target genes. And so we actually, what we do in most instances is we rank the transcription factors, we rank the genes by this differential targeting score. So it's really just, it's like summing up over all the edge weights for the transcription factors and then doing a t-test using lemma. We do a comparison between them and we like lemma because we can correct for some of the covariates since it's a linear modeling approach. But we do that analysis and then we ask, you know, if we rank the genes based on their differential targeting and do a pre-ranked gene set enrichment analysis, what does it tell us? And so that's that's one of the approaches that we use. We've also looked at other network methods, metrics. So one of them is differential community structure, and Mega Patty developed a method she calls Alpaca that does a really good job of finding differences in community structure. Although we had a big debate 2 weeks ago about what actually doing and how to do it better, and a crisis of confidence in everything we've done for the past so many years, but I think we're okay. So the difference in community structure can be really important because what I'd argue is that a difference in phenotype comes because you activate or inactivate a process, right? Not individual gene, but regulation or cell cycle or kinase activity. So looking at community structure and the creation or deletion of communities is a really is a good metric. And community structure is really hard because if I snip a couple edges, I can have two communities separate from each other. Or if I add a couple edges, I can have two communities fused together or any combination of the two. And I always like to use families as examples. So, you know, like I think my wife is Greek, and so she comes from a big Greek family, right? And there was Aunt Pauline, who is the matriarch of the family. And she held the family together. But when Aunt Pauline died, the family broke apart into warring factions because the only thing that kept them together is they didn't want to make Aunt Pauline upset. Okay, so snipping, you know, a couple edges from that one node broke the community apart. In the same way, you can imagine, you know, two families that are separate, but people get married and the families then merge together. Okay, so it's a few edges, a few connections that can change the structure. So community structure is one. The other thing we look at is things like betweenness centrality. So in the paper we published looking at 38 different tissues and looking at tissue specific regulation, what was really interesting was we found that the transcription factors driving tissue specificity weren't at the core of the network. Those were things that were inviolate because they were essential for survival. They weren't at the periphery of the network because they had too little effect. They were things in an intermediate area where they actually gained or lost connections. And they gained connections through what we refer to as non-canonical interactions. So they didn't have the known transcription factor binding sites, but they looked like they were interacting in other ways. And some of this we now think could have been because of open chromatin. So again, I apologize. I always feel bad when I answer questions because I give another whole nother talk, but I'm, hopefully that's okay.
We have another question from Min. Thanks for your great talk. I'm wondering whether these methods are applicable to protein network or metabolic networks.
So that's a really good question. You can, some of the methods we have can be applied to looking at protein-protein interaction networks, although the network inference piece can't. So, looking at differential edge weights is something you can apply directly, and that's something that we've done. Alpaca, this method for finding community structure, it was really, we started developing it using bipartite networks, although you can apply it to unipartite networks. And the great thing about what Alpaca does is it takes the big problem with finding communities is you have to postulate a null model. And what Alpaca does is it looks for, if I have two networks, it takes the network in community A or in phenotype A, and it looks in the background of phenotype B, and then it does the other. So it can find two sets of emergent communities. You can do that if you can define different protein-protein interaction networks, right? And maybe you could do that by imposing differential expression on those networks. It's a little bit harder to apply these directly. For metabolic networks, we've tried some of this, although we haven't had that much metabolic data to try to apply. The community structure detection, though, and actually Panda, we've used to optimize network structure for methylation data, for protein expression data, and for some other data types. But for this regulatory network inference, there's no direct translation. I will tell you, at the expense of getting another way too long answer, is that when we first published Lioness, right, this leave one out linear interpolation method, when we first published that, it was up on archive for more than 2 years. And I was actually contacted by somebody who wanted to use this to look at 3D chromatin structure in cells. So they were doing Hi-C, and they had all this Hi-C data. And what they wanted to do is they wanted to estimate individual cell type or individual cell chromatin structure. So they adopted this idea of basically having a matrix that you estimate from data, you leave a sample out, you re-estimate it, and you interpolate. And they're able to just take the Lioness equation, which is that one line, and 4 lines of code in any language you pick, and implement that to do an iterative interpolative estimate of structure. So that piece you can apply anywhere.
Thank you. And we've been enjoying your answers. You have comments saying as much too. So are there any other questions?
Well, I will say, if you look me up on the Google, I'm easy to find since my name is Quackenbush. Although I noticed I'm starting to get emails now every day from Anna Quackenbush at Illumina asking about their products. And I don't know who Anna is, but I happen to meet her one day. So if you look me up on Google, I'm easy to find. I'm not Anna, although my sister is Anne. She's a veterinarian, so she might not be able to help you. But if you have any questions, feel free to drop me a note, and I'm more than happy to respond to any questions people might have. If you have trouble finding the software, drop me a note. But if that's it, thank you so much, Nadia, for being the ringmaster today. And thank you, John and Min, for inviting me.
Yeah. Thank you so much, John, for the great talk, and I really appreciate your willing to share both the recording and the slides. So we'll soon post it on the workshop website. And yeah, with that, I will thank you all for joining us today.
Nice seeing you all. Thank you so much. And I'll send you the slides.
Thanks, John.
All right. Take care. Have a good afternoon.
You too.
I hope to see you all in person one day soon.
Hopefully next summer, if not earlier.
I miss seeing people and having friends. So we've actually had people over to our house since we're all vaccinated now. So it's nice to start to come back to normal. So bye. Enjoy the rest of the workshop.
Thanks.